Today I'm going to talk about Darwin meeting Freud. There's no social insects in here, at least not yet. I'm going to start with a brief description of conventional medical approaches to psychiatric conditions. I'll describe an evolutionary approach. I'll relate human evolution to psychiatric conditions. I'll present a new conceptual model that I've developed with a collaborator of mine in London, Christopher Badcock, and I'll discuss evidence, tests from the literature and from my own research program of the model using genetic, endocrine, neurological, psychological, and psychiatric data, and then explain at the end why this model and this approach is useful for basic research and for therapy and for understanding our own minds. And the overall structure of the approach is shown here. I wasn't quite sure, Joan, what to do with anthropology, so I put it up there. I know that the circle ought to be, it's sort of swallowed by genetics. I didn't notice that until just now, but I'll work on this slide. This is what I'm trying to do. I'm trying to integrate evolutionary biology with all of these different disciplines, taking evidence from all of them, working across levels, mainly to address the evolution of human social condition and cognition and what happens when it goes wrong. Conventional medical model psychiatric disorders are diseases like type 1 diabetes in principle, but they're defined by DSM-5. Like any other diseases, they represent pathology, something going wrong, caused mainly by bad genes, deleterious alleles, and environmental factors like stress. And to study them, you analyze their proximate causes, the genes, the neurophysiology, the endocrine correlates, the psychology, et cetera, et cetera. This all fine. Absolutely fine as far as it goes. It's certainly part of the picture, but it's not all of the picture. Evolutionary models, with evolutionary approaches, we can look at psychiatric conditions as involving reductions or extremes of evolved adaptations. So with type 1 diabetes, under this perspective, type 1 diabetes, to understand the maladaptation in sugar metabolism, we need to understand the adaptation, how the beta cells in the pancreas work. You understand the maladaptation by understanding the adaptation and figuring out the difference between those two. So we look at the adaptation that is dysregulated. Often these involve tradeoffs as well, and we use a combination of proximate and ultimate approaches. Here I've directly contrasted the approaches conventional and evolutionary, proximate versus ultimate, the human machine compared to the actual evolved human body, connect the maladaptations with the adaptations, develop treatments, prevention. But here, something rather different with evolutionary medicine. We're using the basics of evolutionary theory, the foundation of all of biology, to generate testable hypotheses. Most importantly, it tells you what data to collect that you would not collect otherwise. And perhaps most importantly, the two approaches are fully compatible and should in fact be synergistic with one another. So how are human psychiatric conditions related to human evolution and the adaptations of humans? We have big brains, even Homer. We are especially socially smart. We have a social brain. Much of the neocortex involves this distributed, integrated system for taking in and processing and using specifically social information. And that's epitomized by this fellow here, one of my favorite historical figures. And humans are also, of course, technically smart, because the main way that our world is different from the world a couple of thousand years ago is the changes in material culture. So this in the most general terms, this is what's evolved in humans. We can kind of unpack these general traits and make a list of the phenotypes that are either unique or highly elaborated, highly developed in humans. These would include language, developed sense of self, mentalistic skill, theory of mind, thinking of other people as having minds, highly developed social emotionality, so there's basic emotions, fear and anger, social emotions, pride, guilt, embarrassment, shame, contempt, explicitly emotional in a social context. We have complex long-term regulated goal pursuit, mental time travel involved here, empathic drive to understand and resonate with other people, and of course highly developed visual, spatial, technical skills. And abstract intelligence is intelligence that's separate from the cultural learned aspects of intelligence. So all of these have evolved, and they've evolved in the same way that everything else has evolved, because they must, because that's how evolution works. We have our chimp human ancestor. We've had a series of mutations, many of which have been selected for. Many have fixed, and for many of these traits and for many of these genes here, we have segregating variation in extant populations. So we have all of the phenotypes that made us human, the genes that made us human, and these genes that made us human are still subject to allelic variation that is segregating in normal populations, and we can use this variation to try to understand this whole process. So this is human evolution. How does it relate to psychiatric conditions? If you take all of these social, verbal sorts of traits and dial them down or take them away, what you get is something very, very similar to autism and autism spectrum conditions. Severe autism, no speech, reduced sense of self, lower mentalistic skills, so reduced theory of mind is quite directly diagnostic. Basic emotions are developed, but social emotions, pride, guilt, embarrassment, shame, etc., are not developed, less in the way of long-term goals, and relatively specific effects from reduced empathizing, reduced connections, reduced bonding, affiliation with other people. So we have all the reductions here, but there's also excellent evidence for relative and absolute enhancements with regard to non-social and non-verbal cognition. Many studies showing enhanced visual spatial skills, enhanced technical skills, and relatively high levels of non-cultural, that is purely abstract intelligence in autism. And this way of attaching human traits to autism does fit squarely with the conventional DSM-5 now definition of autism in terms of dialing down sociality and communication and dialing up non-social restricted interests and repetitive behavior. So it is fully compatible with the traditional definitions that came out of Leo Kanner and Hans Asperger back in the 40s. It's very highly heritable. This makes it a special interest to evolutionary biology and evolutionary genetics. Incidence is relatively low, but this is just one extreme. It's a spectrum all the way from severe into normal populations, the so-called autism spectrum. Now, like any other traits, these traits can vary in two directions. That's the nature of biological systems. You have some sort of mean, normality, and they can go in two opposite ways. What happens if you go in the opposite direction for these traits, the opposite direction from autism? What does it look like? Language coming out of nowhere, hyper-developed sense of self, delusions of grandeur, highly inflated sense of self, sometimes referred to as megalomania, overdeveloped theory of mind in paranoia. Other people don't just have minds, but they're using them to think about you and plot against you. Highly developed social emotionality, especially guilt and shame, embarrassment, overdeveloped in depression. Complex regulated goal pursuit, hyperdeveloped, essentially run amok in mania that you find in bipolar disorder, and it's directed towards two of the main historical maximizers of inclusive fitness, which in our society are sex and money. And empathic drive, hyper-developed empathy. I'll talk more about that in a few minutes. One can have levels of empathy and attention to empathy which are pathological. In autism, non-social skills are enhanced, and the opposite direction here is selective reductions in non-social skills. So what we've done is develop a model based on human evolution and perturbations to these major aspects of human evolution that go in two directions and that involve these opposite alterations, essentially trade-offs between social traits and non-social traits. So trade-offs are, of course, fundamental in biology, and I think they're also fundamental in the brain. So what we've described here is what we can call the psychotic-affective spectrum, sometimes called the schizophrenia spectrum. This includes schizophrenia, psychosis, reality distortion, hallucinations, delusions, paranoia, schizoaffective disorder, kind of a mix of schizophrenia and depression, and bipolar and borderline personality is in here as well, overlaps with major depression and bipolar and has some of its own aspects to it. If you saw Winona Ryder in Girl Interrupted, you saw borderline personality disorder. Like autism, these are all highly heritable. So what are the genes for these bad conditions doing? Why are these traits so highly heritable? These all share symptoms, genetic risk factors, and other correlates. They're pretty common, especially for some of them, about 1/6 of individual lifetime at risk of depression. Borderline's up there, about 10% of them commit suicide. Overall, rates of psychosis, about 3.5%. But once again, this is all a spectrum, and these are not discrete, originally described by these two folks back in the late 1800s. Huge costs to societies, individuals, and families. And we can look at this simple model graphically, where we have verbal and mentalistic skills, sort of mind-related skills, empathic skills, social skills along this axis, perceptual, visual, spatial, mechanistic, non-social skills. Systemizing refers to understanding rule-based systems, okay, and as opposed to humans having specific rules like aspects of physics, for example. Balanced cognition and effect along the diagonal. Alleles for autism ought to represent little vectors like this. So if you got one genotype, you might be here. If you got the other genotype, you might be here. And that's an allele for autism. It's an allelic difference correlated with a phenotypic difference that is pushing you over here towards the autism spectrum. So you start somewhere and you add up all the vectors like you did in physics 20 years ago, and maybe you end up here at the extreme of the two axes, low here and high here, and the opposite sort of effects for the psychotic affective spectrum. It's a very, very simple sort of model, and it certainly cannot explain all sorts of cases, but I think it can explain quite a bit. Some of the more interesting evidence for the model comes from a survey given to an incoming class at Princeton, where they were asked what their major was, and also, by the way, what psychiatric disorders are in your family. I'm sure ethics enjoyed that one a lot. People going into humanities, about a twofold increase in depression, bipolar, and substance abuse. This was significant. People going into the technical sciences, about 2.5-fold rates of autism spectrum in their families, showing sort of a pedigree structure to these disorders in relation to the interests of the individuals and also in relation to their scores on psychological tests that rated them for how much they were interested in rule-based systems and how much they were interested in empathic, sort of people-based phenomena. So I think this sort of data is very interesting in connecting up the so-called pathologies with the real world here. Okay, so we have a model The model is based on trade-offs, like so many models are in evolutionary biology and life history theory. The model is based on trade-offs. We don't usually think of trade-offs when we think of the brain. We usually think of things like IQ, which is kind of the opposite of a trade-off because it's many, many traits contributing and all going in one direction on one axis. And it turns out, especially if you look at the literature on autism, there is considerable evidence for trade-offs between social, verbal, and non-social skills. And I've listed most of it that I've come across here. So, for example, verbal skills negatively correlated with spatial imagery skill, empathizing negatively correlated with systemizing. So generally, with autism, it's the kids who have relatively severe social problems who have the relatively good visual spatial skills. So what is the basis for those sorts of trade-offs? Well, one really wants to know because that really is kind of in principle, it's the crux of understanding these sorts of disorders. So we're very interested in what's going on here. For schizophrenia, there's also evidence for trade-offs. There's two components of IQ. Verbal IQ is lower in autism. This is basically nonverbal IQ is higher. We have the opposite effect, relatively high verbal IQ in schizophrenia. And there's data showing that genetic liability to schizophrenia, this is pedigree-based genetic liability. So if your dad was schizophrenic, you would have genetic liability there of 0.5. Genetic risk of schizophrenia is quite strongly related to having better verbal skills compared to spatial skills. So the disparity between verbal and nonverbal skills is very strongly related to liability to schizophrenia. Once again, we're going into the corner of that plot that I showed you just a minute ago. So this is our model. It's a nice evolutionary model, but it's just a model. It's just a hypothesis. We need some real data to try to test this. I've been trying to do this for the past several years using literature data and data from my own lab. Today I'm going to present some data addressing some of the main predictions. I'll first talk about opposite phenotypes in autism, and mainly the evidence is from schizophrenia. That's because most of the work has been done on schizophrenia, a lot less generally that's relevant for depression and bipolar. So we'll look at more specific phenotypes in these two conditions, and then we'll look at the diametric genetic basis, which is a critically important aspect of the model and tying it into evolution. There's two types of genetic alterations where the alterations are big effects, and they can go in two opposite directions, and they involve human social cognition. And these are copy number variation and imprinted gene syndrome. So these are absolutely beautiful natural experiments where the human genome has gotten perturbed in some people in one direction, in other people in the exact opposite direction genetically. So it's just the sort of test that you would want for the model. And then I'll discuss some of the results that we have from my research program. Are there diametric phenotypes for the two conditions, autism spectrum, psychotic-affective spectrum, high birth weight in autism, low in schizophrenia? This has now been shown clearly in the same populations, ties into large versus small brain size. There's amazing data from Asperger's syndrome showing an absolute lack of loss of gray matter with age, and I want to know why that happens because that's a major aspect of cognitive decline, of course. And with schizophrenia, this is greatly accelerated loss of gray matter. Large, more reactive amygdala in autism, smaller in schizophrenia. These are some social brain regions, Brodmann areas. These regions are less activated, showing less engagement in social cognition in schizophrenia, but high levels of activation in schizophrenia, although it's a little hard to interpret just what hyperactivation means. Reduced activation of the daydreaming network, the resting network, when you're sitting and not doing anything except thinking. This is the default network that the neuroscientists just discovered existed about 10 years ago, and going crazy about increased activation of this network in more regions in the network. These are references. A lot of it is collected in this 2008 paper and some more recent papers of mine. Reduced mirror neuron system activation in autism. So this is the brain system that's involved in resonance between individuals. So when you see me moving my hand, you're actually activating some premotor neurons that you would use if you were going to move your hand. If you're engaging in your mirror neuron system, it's kind of a mental neural mirroring between people. This is reduced in autism. People who are actively in psychosis show increased activation of that system. We've talked about loss of speech. We've talked about verbal and visual spatial skills. We've talked a bit about empathic skills. One of the most remarkable findings to come out of this whole body of relevant work is that if you want to look for enhanced empathic skills in humans, which sound great, don't they? Wouldn't we all like to be more empathic? You know, think Mother Teresa and everything. Where do you look? Do you look at politicians? Well, maybe. Do you look where you should look, where the evidence says you're going to find it? Borderline personality and mild depression. These people are better across a whole range of empathic skills. For example, being able to read emotional state just from looking at people's eyes. People with borderline personality will score better at this. Their lives are a mess. They have huge mood instability, disrupted sense of self, impulsivity, depression, psychotic episodes. They have highly increased social attention. So it seems, and this seems to be associated with better empathic skills. And just how are they doing that? Better local relative to global processing of information. So they're better at finding Waldo on the page. This ties into increased short-range brain connectivity, at least in principle. Evidence for the opposite sort of effect in schizophrenia. Hyperlexia, the precocious untaught ability to read really fast, is tightly linked with autism, and dyslexia is actually quite closely associated. This is difficulties with reading, quite closely associated with aspects of schizophrenic cognition. There's connections between these. Increased fluid intelligent abstract intelligence, working memory processing speed, in autism, found in a number of studies, but fluid intelligence, working memory in particular, decreased in schizophrenia. Bipolar is quite different. That's pretty much another talk. Attention to gaze, inference of intention, shared attention, personal agency. We can think of these as all kind of uniquely highly developed in human traits. If we increase gaze sensitivity, this has been found increased in paranoid schizophrenia. Inference of intention, hyperdeveloped delusions of persecution, erotomania, that's pretty much my favorite psychiatric condition. It's what it sounds like if you know Latin. Well, maybe not. You believe that somebody who's really famous and important is hopelessly in love with you. And it's a particular, very interesting delusion. Shared attention is reduced in relation, and if it's increased, delusions of conspiracy, and we mentioned megalomania. Just a few more. Part of the autism spectrum involves major deficits in ability to lie. They're not good at lying. This has got to be a major sort of social interaction problem for them. This is in contrast to the psychotic-affective spectrum. One of the subscales, diagnostic factors, involves supernatural thinking, magical ideation, believing in sort of magical events. Reduced imagination in autism, often seen in reduced symbolic play as a child, so they don't, pick up the little figures and say, Doody did this, and then they went over there and did this together. Instead, they'll just line up the blocks, for example. But there's a whole book written about strong imagination, enhanced imagination in psychosis by Dan Nettle, and it ties in with a large literature on people that I refer to as psychotic savants, especially in arts and literature and poetry, people like Sylvia Plath, who were psychotic, and when they were not actively psychotic, they were producing some of the most amazing literature that's ever been written. Repetitive behavior in autism, resistance to change, don't move around the furniture. Psychotic-affective spectrum, they're essentially taking in everything. There's the opposite of repetitive behavior is very sort of loose associations, holistic thinking, coming up with new ideas, novelty seeking are all tied in here. We can summarize all of this literature with a quote from Bill Hamilton, my thesis advisor from years ago. People can be divided or stretched between two extremes, things, people, and people. So that's phenotypic evidence. The main problem with it is, it's fine as far as it goes, but there's not a lot of studies that have looked in the same study with the same protocols at both autism spectrum and psychotic-affective spectrum, and that's what you really need. That's the data that needs to be collected, that evolutionary biology is saying, go and study that in this way. It's certainly been done in some cases, but not in all that many. So what about genetic variation? I'll first talk about copy number variation, called CNVs. These are regions of the genome, anywhere from 1 gene to like 50 genes. Most people are normal. Here's your 2 chromosomes for that chromosome number, and you've got 2. Sometimes a big chunk of DNA will just basically get hacked right out, and you'll be haploid for a great big region, and sometimes that exact same chunk will be stuck in where it shouldn't be. So you're triploid for that region, okay? So you're either haploid or diploid or triploid for the exact same set of genes. You have 2 alterations, and they're exactly in the opposite direction. So what we would predict would be when you go one way, if you got autism more one way, you should get schizophrenia more the other way, although the actual genetic cause that's underlying it is generally not well understood. I pulled all of this evidence together first in 2010 from all of the CNV studies, and then again in 2012. And there's a total of 7 loci where you can do this. 4 of them do show the expected patterns where deletions are associated with one disorder and duplications with the opposite disorder. So these findings are, I think, quite strong support for the model, and even stronger support comes in particular from this particular CNV, chromosome 22, the Q arm, band 11.2. This is called velocardiofacial syndrome. It's one of the most common copy number variants, and it involves deletion and duplication of this same region. A paper just came out a short time ago showing that duplications of this region actually protect against schizophrenia. If you have deletions of the region, strongest known risk factor essentially for schizophrenia, about 21-fold increased, okay? Roughly a quarter of the people who have it will get schizophrenia. If you duplicate the region, you have increased risk of autism, also very well established statistically. That's basically what I just showed you. But people with this duplication and many of them do not have autism. Most of them do not have autism. People with that duplication are actually protected against schizophrenia. They show schizophrenia at roughly 1/6 of the expected rate. And this is actually the first major protective mutation against schizophrenia, and it's something that actually causes autism. And you want to know what these things are, because this could point you directly towards therapies that would prevent schizophrenia in people who are at risk. So there are very direct therapeutic implications in this case, and there should also be in those other sorts of cases that I talked about. There's very specific predictions about what data you want to collect. So that's copy number variation. The other situation where you have these big perturbations in 2 directions is imprinted genes. Normally, for imprinted genes. One of the copies is imprinted. That means silenced, turned off. You don't express the gene. You can have alterations where the gene is unsilenced, so you got double expression in principle, or the other copy is silenced, where you have no expression, and that's normally very bad news developmentally to lose expression. These are usually pretty important genes. But for our purposes, we have large perturbations in two opposite directions, and so we have very clear predictions, and the predictions come out of what's called the kinship theory of imprinting, developed by David Haig at Harvard University. And the basic idea here is that humans have a history of some degree of multiple paternity, and that means that alleles that are inherited from the father, paternal alleles, will show lower relatedness in a brood. So think of a bunch of kids, each has a different father. For paternal genes, they're related by 0, okay? They might as well be strangers. They're not going to be selected to be nice to one another in this little thought experiment, and they're going to be selected to each want to take more from the mother, okay? They don't want any resources going to their siblings. They gain no relatedness benefits from that. So we expect paternally expressed genes, maternally silenced genes, to be involved in increased demands on the mother. Maternally expressed genes ought to show the opposite here. So selectively, what is going on? Fathers are shutting off their genes. When they make the gametes, they're shutting off the genes that reduce demands on the mother. Okay, so the genes that are expressed are the ones that increase demands on the mother. Those are paternally expressed. When the mother makes eggs, she shuts off the genes that increase the demands on her, and that results in the maternal genes that are expressed are ones that reduce demands on the mother. So you have conflict in the genome. It's very counterintuitive. It's very strange, but it's a very basic prediction from evolutionary theory. Imprinted genes are especially important in growth, major effects on baby weight, birth weight of babies, also major effects in brain development. Here's a little example of this sort of genomic conflict within a fetal mouse. Right in that one mouse there, you have paternal genes making a growth factor. Some of the growth factor binds to these receptors to stimulate growth. Some of the growth factor binds to this decoy receptor, it's called, and just gets broken down. Okay, so you're cranking out this critically important growth factor, and a large chunk of it is just immediately chopped. Physiologically, it's absolutely, completely stupid. Makes no sense whatsoever. It's a tug of war, and it creates a conflict system that makes opportunities for all sorts of dysregulation, at least in part because these genes are haploid. Okay, so they're very vulnerable to mutations and alterations. So what about the brain? That's fetal development. We have a lot of imprinting effects in the brain. Some early work by Barry Cavern at Cambridge showed he made mice that had mixtures of normal cells and either sort of all maternal cells or all paternal cells in their brain, and then he looked at the fates of those cells, and what he found is that the maternal genes, with 2 copies of the maternal genes, tended to proliferate more in the neocortex, and where you had 2 copies of the genes from the dad, these tended to survive and proliferate in the hypothalamus. And the way I like to look at this is, this is a very sort of broad distinction, okay, about as broad as you can get. This part of your brain says, I want a milkshake. I really want a milkshake, okay? You can't even talk, but you want a milkshake. And this part of the brain says, no, maybe you really shouldn't have that milkshake. It might taste good, but it's not really good for you. It's full of sucrose, yada, yada, yada. And somehow you sort it out and you have the milkshake or not. You have basic drives in your life, and these are basically coming from this part of the brain, and then you have to think about how those are going to work and how are you going to apply them. Yeah?
Is that a requisite of Canadian research?
it is good to try to work it in. You can get sort of these fundamental insights. We're the only species that has group against group competition in play, as Dick Alexander is always very fond to point out. And I think that tells us something quite interesting about humans. And hockey is kind of the epitome of that. It's kind of the closest to warfare of all of the sports, as far as I know. So the basic idea here is that we have these paternally expressed genes that are having effects in the hypothalamus and these neocortical effects on higher functions, according to what Cavern found, and that's all well and good. For me, the particularly interesting aspect of it, for those of you who have read Freud with an open mind and didn't just kind of throw it out the window because of all his excesses about various and all of the jokes that have been made, is that Freud developed a theory about conflict in the mind between basic drives, basic instinct, id, the pleasure principle, and the superego or moral conscious. These are in conflict, and then the ego is basically kind of sits in the middle and, well, what are we going to do here, trying to sort it out. And this was, of course, completely independently developed, but it does map on surprisingly nicely to this sort of framework. And both the evolutionary approach and Freud's approach, which has developed through the years into psychodynamic, psychotherapeutic approaches that work, if I could say that, Randy, at least some of the time, is that both of the theories center on the strong effects of early child development. Anybody doing child development is going to tell you about the strength of the early effects. Social relationships with parents are fundamentally important in both cases. Conflicts in the mind, mental illness coming from imbalances between the competing forces. So there's this sort of remarkable sort of convergence here between these viewpoints. And one can look at it in this sort of balance model that we can then tie into the genetic data. So here we have balance between maternal and paternal effects, sort of a tug of war. Okay, it's a dynamic tug of war. There's pulling going on. There's not canalization. There's not homeostasis. There's not negative feedback. Well, to some extent there is, but then there's this opposite force, which is set up for failure, essentially. If you gain paternal gene expression or lose maternal gene expression, you go this way, and this paternal brain bias, I'm expecting to be involved in the autism spectrum, the social brain underdeveloped, and an opposite where you get a maternal brain bias, then the maternal brain overdeveloped. And one can see this with relation to our earlier model. These are all the social brain traits. They're all maternal brain traits. A lot of them probably developed quite squarely in the context of mother-child interactions. And if you dial those up, this is what you get, this excessive imbalance of influence towards the maternal brain. So this is how we can tie it in with imprinting effects. So what does the data look like? Once again, we have the perturbations in two directions. I'll just talk about a couple of imprinted gene syndromes, once again, that go in opposite directions towards excess paternal compared to excess maternal. If we have an alteration on chromosome 15, so we lose maternal expression, and we're going the paternal way, this is Angelman Syndrome. They're very demanding as young children. They cry all the time. They stay up half the night. They want to suck all the time, even if they're not so good at it. Very demanding as young children. Every mother's worst fear as far as child behavior fits in with increased demands on the mother. Very high rates of autism. Same region, imbalance towards maternal expression. They're really easy as a child. They just lay there. They don't want to suck. They got to be tube fed. They don't cry. They sleep all the time. Kind of the extreme of what people tend to want, an easy baby. But when they grow up, they have some of the highest rates of psychosis for any sorts of disorders. We also have evidence from this overgrowth and undergrowth pair of imprinted gene syndrome, spec with Wiedemann, paternal bias, overgrowth. Kids are really big, come out at 10 pounds, develop a lot of childhood cancers. Really big tongue in particular. The tongue is the suction pump for getting milk from the mom, and that's soliciting resources from the mom. Very small tongue in Silver-Russell syndrome, but with Wiedemann, very high increased rates of autism. No psychiatric studies of Silver-Russell in particular, but low birth weight is quite strongly associated with higher risk of schizophrenia. There are a variety of specific genes that show parent of origin effects or imprinting effects. I'm not going to go into the details here except to say that for all of these genes, if we know the direction of expression, we can predict what we expect from the perturbation with regard to how they should affect the brain and how they should affect these disorders. And some of these genes also show quite interesting links in terms of evidence of positive selection in the human lineage. So you can link up human evolution with the perturbations that give you the different disorders and kind of wrap up the complete picture. So I'm going to spend a few minutes now talking about data from my lab. For about the past 4 or 5 years, we've been collecting data to try to test these models because so far nobody else is, and I want to see the data. We collected data from non-clinical populations, which is a euphemism for undergraduates. We're up to about 50, I didn't say healthy populations. We're up a bit over 1,500 Caucasians, so-called neurotypicals. We are genotyping them for relatively well-validated SNPs. So for the first time, we have good evidence, good autism risk and schizophrenia risk genes. We didn't have that even 3 or 4 or 5 years ago. We've got it now. The evidence is good enough to say this is an autism risk gene, this is a schizophrenia risk gene. So we're looking at those. What do they actually do? We're looking at hormone-associated SNPs, specifically oxytocin and testosterone, where you can predict serum levels from sets of SNPs, which is very exciting. We've looked at a few imprinted genes, both SNP data and some methylation data. We're measuring autism spectrum and schizotypy spectrum with questionnaires. This sort of gives us some idea of where people are on this spectrum with normal in the middle. We're looking at some visual spatial tests, some verbal skills, and various other metrics, handedness, empathizing, and systemizing. And I'll just have time to talk about some of these results today. But what's most important in these data to do initially is say, can we pick up an axis? Does this axis actually exist in normal populations? And to do that, we did some kind of old-fashioned psychometric sort of factor analysis, which you do with these sort of questionnaire data. We gave the autism spectrum and the schizotypy spectrum questionnaire to large numbers of individuals, and you get your correlation matrix, and you factor it to see what the sort of higher level structure of those data are. And you get your principal components axes. These are the 2 major axes of variation in the data. These are each subscales, sort of components of these spectra as measured with these standard ways of quantifying them. So, and then you look at the size of these loadings here, the magnitude and the positive or negative nature of the loadings, and then you combine that with the biology to say, well, does this seem to represent anything real or biological? Principal component 1 loads most heavily on the socially related subscales, and it appears to represent an index of reduced general social functioning and interest. And we think we can interpret this in part as kind of an effect of these sort of psychiatric variations. So if you're psychologically different, and one of the effects of that is sociality, social interaction, social interest, social skills tend to be reduced. I mean, social skills and social interest are reduced in schizophrenia. I mean, if you gave people with schizophrenia an autism spectrum test, they would test as autism, but all you're doing is picking up deficits. Okay, and you can have deficits in vision from being mostly blind or hypersensitive to light. So you can have opposite effects that opposite causes that give you something that looks like the same effect if you're measuring it as a deficit, which people in these fields tend to do. And we want to try to look beyond deficits. So PC2 is what's of interest here. This is an axis between autism and what positive schizotypy. These are the traits that kind of emerge, the new traits in schizotypy. We have the strongest positive loadings on these positive schizotypy traits, and these show opposite loadings to the main components here of autism. And we can validate this by looking at PC2 in relation to handedness. Mixed handedness is a good correlate of schizotypy and schizophrenia, and it's correlated with mixed handedness and probably reduced lateralization. It's sort of an independent validation of PC2 as an index of an axis from schizotypy and psychotic-affective conditions over to autism. So we've got something we can measure, sort of psychological data that gives us some purchase as regards quantifying people along this axis. And one can look at these axes this way. Here's PC1, and then here's PC2, and we're talking about where people are along this particular axis. I should mention that this pattern of loadings, magnitude of loadings, has now been replicated in populations in Japan over in Italy. So it really does seem to be something which is out there and it seems to be quite fundamental in terms of the correlation structure of these tests. Is it genetically based? For 114 SNPs, most of them autism risk, schizophrenia risk, or endocrine-related SNPs, about a quarter of them are significantly associated with PC2 score, which we think is quite a large number and suggests that there is a genetic basis to PC2, which is what one would expect if it's really fundamentally biologically based and involved in the actual disorders that we know are highly heritable. And this includes, these associations include SNPs in some of these genes which have been linked to both autism and schizophrenia, so the predictions are very clear there. And the only SNP that's been associated specifically with psychosis. And we're extending these results. We can do direct tests by looking to see how specific SNPs are related to autism and psychosis traits, just to see if there's opposite patterns in the genotypic association. So that's a very precise expectation there that we're coming up with next in our program. That's genetic data. Genetic data is good as far as it goes. What's kind of in between the genes and the psychology and the psychiatry? Well, one of the phenotypes that's right square in the middle and we know is important is hormones, genes, neurodevelopment. There's hormones. What do? They motivate cognition and behavior, and they modulate tradeoffs, fundamentally important in social affiliation and all sorts of behavior. Just in the past few years, we have the first genetically based indicators of serum hormone levels. So we can genotype a set of SNPs and look at somebody's genotype and say, oh, you've got a set of genotypes for these oxytocin-related SNPs that say your serum oxytocin is expected to be high, and it's probably been high for your entire life. Okay, we don't know that yet, but it'd be very surprising if it wasn't. So this is, to some extent, it may be more useful than just measuring plasma oxytocin because it gives you a genetically based measure, and genetics are fundamentally causal. So we've got these sort of SNPs for oxytocin and testosterone, which are especially interesting ones to look at because there's a lot of evidence showing that they have opposite effects for quite a range of phenotypes. This is administration studies. You give people hormones up their nose or quantify them with blood or both. These hormones have opposite effects on trust, cognitive empathy, parental behavior, your scores on reading emotions from the eyes, and perhaps most interestingly, they have opposite effects on connections between the limbic brain regions, the paternal brain, here the amygdala, and the maternal brain region. So the amygdala is sitting down here generating emotions. Okay, you ought to feel this emotion here. And then the maternal brain is sitting up here. Well, what am I going to do with this emotion? What's the best thing to do with this? How should I behave as a result? And it kind of goes back to our maternal and paternal sort of axis, although in a more general way. So these are having opposite effects and interacting with one another. Does this tie into the axes that I've been talking about? Here, red means negative correlations, blue means positive correlations. We have very big sample sizes. These correlations are pretty small, okay, but because the samples are big, they're significant. If you have high serum oxytocin, you're not as good at mental rotation. You got a better imagination, and you have a higher score on PC2. Positive schizotypy is higher, autism relatively lower. That fits in with a lot of the work on oxytocin and autism. For testosterone, once again from 3 SNPs, higher testosterone, you're less likely to believe in the Easter Bunny. You have lower cognitive perceptual score. You don't believe in UFOs, and you have a lower score on PC2. And you can put these together based on the model that they're antagonistic, and then it all kind of comes out in this metric of testosterone relative to oxytocin. It's based on only 6 SNPs, which is quite amazing to me. We're explaining very little of the variance here genetically, but we're still getting effects which are pretty much completely compatible with previous studies, which is quite exciting. So higher testosterone relative to oxytocin, higher mental rotation, lower imagination, lower on the Easter Bunny, higher total schizotypy, and lower on PC2. So there seem to be hormonal associations here that can once again give us some sort of purchase and help us tie this in with all of the wonderful work going on, especially on oxytocin these days. And we can look at these results graphically here. Serum testosterone in relation to oxytocin. Once again, higher, especially positive schizotypy here, and then over here, autism, and that's some of the corroborating evidence. And this ties in at least at a superficial level so far with Baron Cohen's extreme male brain ideas, which go back to Hans Asperger. Oxytocin is also known to direct attention towards social stimuli and away from non-social stimuli, which is probably a major factor in a lot of these conditions, selective attention determining what goes into the brain in the first place. What about non-social visual, spatial versus verbal skills? I'll show a little bit of our specific SNP data with regard to these different types of skills. We have a couple of SNPs now, autism risk SNPs, where if you have more autism risk alleles, you have better mental rotation ability. So to the extent that this is a general finding, and there is some evidence out of Baron Cohen's lab where they get the same results as us for some of these genes, then we really do have a trade-off going on between better visual, spatial, higher autism risk. Once again, social, non-social trade-offs. By contrast, for schizophrenia, we have data showing for a polygenic risk score that's based on 32 of the best validated schizophrenia SNPs. If you have higher genetically based schizophrenia risk, you have lower mental rotation ability, at least for males, for some reason. So it's the opposite sort of effect. And ideally, what you're testing for is a direct trade-off. We do have evidence for these direct trade-offs. This is one of the best validated schizophrenia risk genes, D-amino acid oxidase, this particular SNP, very well studied in our data. It shows a direct trade-off. If you have more schizophrenia risk alleles, you score better on this simple verbal test of vocabulary, and you score worse on mental rotation, and both of those are significant. So what's going on in the brain? Then if this is a real trade-off, it needs replication. If it is a real trade-off, well, then what's underlying it? Okay, that's what we really want to know. We've looked at a couple of imprinted genes. Schizophrenia risk alleles do tend to associate here with positive schizotypy, here with positive schizotypy, and total schizotypy. We're also looking at promoter methylation in this particular gene and picking up effects. This is not imprinted gene-related methylation. This is just methylation that gives you an indicator of degrees of expression, because if it's more methylated, you expect less expression. And we're going to be looking at more of these imprinted genes. And we are able to link them to some of these measures of schizotypy. So suggesting that those genetic conflict effects are going on in people like us. Okay, what's actually happening? How does it play out? It's a little early to say. So I've talked about a model for relating autism to psychotic affective conditions. It's inspired by evolutionary biology. We talked about some diametric phenotypes in autism and schizophrenia. We talked about genetic basis, large-scale effects from copy number variation in imprinted genes, and some smaller-scale effects from my work on non-clinical populations, which we're expanding in as many directions as we can right now, and trying to tie in with other research groups to test these ideas in a bigger way. So finally, what does all of this tell us about basic research? What does it tell us to measure? What should we do? Why does it matter? Does it help us understand ourselves? What are the actual on-the-ground applied implications of all of this? Well, I think first, and perhaps most importantly, it gives you a new framework for understanding human social cognition and how it ties into these disorders. Historically, people who work on autism don't work at all on schizophrenia. Almost totally independent lines of research ever since the beginning. But to the extent that this is true, you can get reciprocal illumination of each of those research areas on the other. If you find something in schizophrenia, you should say, I wonder if the opposite sort of effect is found in autism. Okay? It tells you specifically and directly what you should measure. It gives you leverage in using the information that you have and in designing studies or collection of data. What about therapies? As far as non-pharmacological therapy, we can use evolutionary tools and perspectives with regards to so-called talk therapy and understanding individuals in terms of their social cognition in the context of where they sit along these axes. If they're relatively autistic, then you want to get them to be relatively psychotic-affective. If they're relatively psychotic-affective, you want to push them towards being relatively autistic. Okay? And it's a little bit provocative to say it that way, but you're just trying to get them more towards the middle where they will be happier and have better social functioning. It gets even more interesting when you talk about pharmacotherapy, because there you have very specific predictions, and there is quite a bit of evidence now that I've been gathering over the years showing that if you have agonists, for example, for some receptor that are used or proposed for one condition, you should think about using antagonists for the same receptor for the other condition. And there's evidence from suggested studies and ongoing studies and clinical trials and drugs that are in use now for 4 different receptor systems for these effects. Okay, Fragile X syndrome, for example, one of the major autistic syndromes, the mGlu5 activation is constitutive. It's overactivated. You want to dial it down, and independently, the people working on schizophrenia are trying to dial it up. Okay, and what should you do to see if the drug is working? Well, you give them the drug, and then you give them a little questionnaire: how autistic and how schizotypal, where are you on the axes? Are you better at finding Waldo? There's very, very, very specific insights about ways of developing pharmacotherapies and ways of testing to the extent to which they're working. What about understanding ourselves? To the extent there's conflicts in the brain, it should generate ambivalence and indecision and perhaps anxieties, and being kind of a natural aspect of human existence. and tradeoffs as being a fundamental feature of human cognition, I think is tremendously interesting and has simply not been used in psychology or psychiatry. These can all be seen as having natural, evolved biological bases. And really, this all represents human cognitive diversity or neurodiversity. And it's a little bit like cultural diversity, and it's a very different viewpoint about psychiatry and psychology, and particular mental conditions not being seen so much as diseases, but being seen in the context of the evolution of human social cognition and what happens to it, and both positive and negative features of all of the different sorts of conditions that I've talked about. Thank you very much. Athena.
It seems like there's two potentially slightly different things going on here in terms of why you have this diversity. So one is just that, maybe there's some sort of mixed strategy where it's good sometimes in an environment to have more social skills, and it's good in another environment to have more technical skills, and maybe there's some, developmental switch to that may be at play. But then there's perhaps another component, which is one that's more related to sort of responding to the demands of that particular moment or environment or life stage for a given individual. So I'm thinking of you put testosterone and oxytocin up there, and I know you're sort of talking about the genes underlying them, but we all know that testosterone and oxytocin are heavily affected by social inputs. So those could be responsive to the particular conditions that an individual is facing, and you could be moving around in terms of that space. So I'm wondering if maybe you could just sort of speak to how you see these settings as it's sort of genetic or developmental or response to immediate environment. How do those things interplay, and what are the most important aspects?
Yeah, well, how much plasticity is there? I mean, that's especially important in something like autism. Certainly it's been shown that if you get autism diagnosed early enough, there really is enough plasticity that you can have major sorts of advances there. The problem with schizophrenia, just because it doesn't develop until 18 or 20 or whatever, the brain is pretty much fully developed and to some extent kind of overdeveloped, so that horse is way out of the barn. The degree to which there is plasticity and gene by environment interactions here and how those would be expected to vary, those are really open questions. One would expect gene by environment interaction effects to differ depending on where people are along these sort of axes. I mean, I've tended to think that there really are. I mean, I see the strong heritability and the big genetic effects even from a particular SNP, and I go, wow, it's all genetics. But then one thinks about the extent of plasticity of the brain, and and the large body of neuroscientific research that says, it's plastic. It may be plastic enough that you can move people long distances in sort of optimal directions, via combinations of talk and.
What about sort of the issue of responding to immediate environment? So I'm thinking of sort of things that we think of more in the domain of like psychology and behavior, like higher testosterone because you're in male-male competition mode, and it's actually good to be less empathetic too. So that kind of level of responsiveness to the immediate environment.
Well, certainly one would expect the degree of any sort of social plasticity to be reduced towards the autism side. One could also think of the opposite of that being sort of hyperplastic. And that could tie into certainly to some of what you find in schizophrenia in terms of their sensitivity to, it's kind of hypersalience. Okay, so if I see something like this strange black box here, and I'm not quite sure what it does, then it becomes very important, and I start to develop ideas that are focused on how this thing is controlling my thoughts, and that's kind of like hyperplasticity in terms of I'm too sensitive to certain aspects of the environment and make too much of it. So that that plasticity could be another very important dimension here. It's one that I haven't thought about. Very interesting. Yeah.
I don't see why the maternally biased brain should have all those kind of properties that you associate with it. Like, what's the, is it an empirical, I mean, you seem to suggest it's an empirical claim, but what is the theoretical
Yeah, that's a really good question. Well, I mean, that's what the major association areas of the neocortex, the classical social brain, has all of these different areas that are specialized for different functions, and those functions can be seen to a considerable extent, I think, as involving sort of regulation of basic drives. And I'm kind of almost going back to Freud here. I mean, inclusive fitness theory, what does it tell us to do? Well, maximize your inclusive fitness, okay? Get money, get mates. Not necessarily be aware that you're doing that, because that's probably not the best way to do it. But that's the basic long-term goal. That's what evolution does. It selects for maximizing inclusive fitness, which gets down to reproduction and survival. But then all of the neocortex, the maternal brain areas, are involved in sort of the strategies involved in doing that, and mainly social strategies involved in doing that. And those develop in social contexts. And what's the main social context of an individual in their whole life? It's their mother. It's the first 2, 3, 4 years of life, your environment, and then before that, totally your mother. Your social environment, to a very considerable extent, is your mother. And so the maternal brain can almost be seen as an extension of the mother's brain and the mother's influence into the child. So the mother has this kind of a big advantage in terms of indoctrinating you into, good behavior, proper social behavior, which should be behavior that benefits her more than you, okay, because of parent-offspring sorts of conflict, because you're genetically half different. I hope I'm getting somewhere near the answer to your question.
I'm just wondering why, because this would mean that the paternal side is somehow maybe suppressing genes that express the social and moral and self-restraint side. What is the advantage for, I mean, I see it in the context.
There's no advantage here. You don't look for advantages except with regard to the transmission of the particular parentally inherited genes. The system makes no sense adaptively in the context of the individual organism. That's the strange thing about intragenomic conflicts. And it's not just imprinted gene. This is conflicts between genes within a single so-called individual. It's not individual anymore. There are conflicts. How do they play out? There could commonly be kind of a tug of war, an intermediate optimum. There could be somebody who wins, but there's no adaptiveness to it for the individual as an entity.
Yeah, there isn't something adaptive to them. So this suggests that somehow mothers prefer their children to have these set of traits, and the fathers prefer their children to have the other set of traits. And that I don't see the rationale for that.
It's not necessarily, I mean, there are those direct influences, but the influences of the particular genes are expected to occur in the context of soliciting resources from the mother, both energetic resources and psychological resources. So the paternally expressed genes in the kids are having effects. Take more from the mother. Take more energy from the mother, get more milk from the mother, get more attention from the mother, get more. And then the maternally expressed genes are, in that same individual, doing the opposite.
Share with your siblings.
Right, exactly. Be nice to your brother. Stop whacking your brother.
Do these patterns of maternally and maternally imprinted genes recur in other non-mammals? Nice way to test that idea. So birds,
Well,
say,
most evidence,
I don't care.
yeah, most evidence is from mice. It's been best studied in mice. People are going, there's data from horses, there's data coming out in chickens and honeybees. It does seem to be going on. I think there's evidence from, initial work, it's not out yet, from birds, they're social birds, biparental. One would expect those sorts of effects. In other primates, the patterns of imprinting do tend to differ between taxa, so they're conserved, maybe 70-80% between humans and mice, for example.
It's the opposite of the mammal. Outside mammals, you might expect quite different patterns, and that would really allow you to test that idea that there is.
Or sex-reversed care.
Well, plants as well. Plants are tremendously interesting because they have genomic imprinting totally convergently evolved, of course, and it centers on the endosperm. And that's the investment, and that's what people eat. If you want to modify plants to make more food, then you want to understand genomic conflict in plants, because the size of the endosperm is a consequence of that conflict.
So where does sort of non-transmissible epigenetic modification, so not imprinting, but changes, lifelong changes in gene expression? So one thing's Michael Meany's study with the parenting behavior producing different stress responses through modifications of the corticoid receptor that lasts throughout life.
Yeah.
This recent study in PNAS that showed that the critical window for in which poverty influences adult ability to do emotion regulation with deficits, enhanced amygdala activation and a decreased medial prefrontal cortex activation with those that were exposed to extreme poverty. So, in many ways it's getting at the same thing, but we're not looking at SNPs. I don't know how else to explain it. It's environmental and it's probably epigenetic.
Yeah, well, that gets into these ideas in, I think, the most general and fundamental way, and that's the idea of trade-offs, the importance of trade-offs and the importance of conflict. So what's going on with those, the licking and grooming? Well, yeah, maybe the mother is preparing them for what's coming ahead, or maybe the mother is doing something that has effects that are in her interests a little more than in the offspring's interests. With regard to other sorts of effects of early development and adversity, I would want to know, I would want to try to measure trade-offs and say, okay, why do we get this sort of alteration? Are there trade-offs there, or is there compensation? To what extent is it pathology? To what extent is it compensation? To what extent is it actual trade-offs involved, such that there's benefits in one context but costs in the other ones?
So if that's then in an evolutionary context where we expect changes in gene sequences, does that mean then that there is a difference in the likelihood that certain genes would be modified in certain individuals? That there's a genetic difference in the likelihood to have epigenetic modifications? In which case I didn't know what.
Yeah, well, the question is who is in control of the epigenetic modifications? There are big influences of the DNA sequence on the epigenetic modifications. These have been shown in mice, for example. But then the question becomes, if there is intragenomic conflict, then what agents would be in control of methylation and gene regulation? It may be that conflict is not that important because there's also selection to reduce the costs of conflict as well. But that's the other main dimension that comes out of evolutionary biology, is when you see a phenotype, who controls it, who benefits from it, are there conflicts, and are there trade-offs? And when one is analyzing the proximate the proximate mechanisms, these questions are commonly not raised. So, the idea of who controls methylation, this is an area of very active research for genomic imprinting. Some people think that the mother is running the show in imprinting establishment, but there are ways that paternally expressed genes can get around that. So it's conflict. It's very interesting because it's molecular conflict. It's antisense RNA. It's decoy receptors. It's molecular biology, but it's also evolutionary conflict. So it's a really neat area.